Welcome to this lecture on the endogenous antigen processing pathway. Today, we are going to explore one of the most fascinating and crucial security systems inside the human body. We will learn exactly how our cells act as internal checkpoints to detect hidden viruses and even cancer. By the end of this video, you will understand the step-by-step biological machinery that keeps you safe from microscopic threats. To understand this pathway, imagine a busy manufacturing factory that has secretly been taken over by a rogue manager. The factory looks completely normal from the outside, but inside it is secretly building dangerous weapons. How can the police outside know what is happening? The factory has a special security protocol where a tiny sample of every single thing made inside must be displayed in a glass window facing the street. If the police see a weapon part, they destroy the factory. In your body, your cells are the factories, viruses are the rogue managers, and the endogenous antigen processing pathway is that exact security protocol. Now that we have our factory analogy in mind, let us look at the biological reality. This pathway is a highly regulated cellular mechanism used to present antigens that originate from inside the cell. When a cell is infected by a virus, or when it undergoes a cancerous transformation, it begins synthesizing foreign or abnormal proteins right there in its cytosol. Because these proteins are made inside the cell, we call them endogenous proteins. The immune system's antibodies cannot simply cross the cell membrane to see what is going on inside. Therefore, the cell must have a way to actively process these internal proteins and push them to the outside. Understanding that these foreign proteins are trapped inside the cell, we must next explore how the cell prepares them for display. The first step is identification. These endogenous proteins are tagged with a specific molecule called ubiquitin, which essentially acts as a biological death sentence, marking them for destruction. Once tagged, the protein is directed to a large cylindrical protein complex known as the proteasome. You can think of the proteasome as a highly efficient molecular shredder. It takes the long, folded viral proteins and chops them up into much smaller, manageable pieces. These resulting pieces are short peptide fragments, usually between 8 and 15 amino acids in length. With the proteins now shredded into small peptides, the next challenge is moving them to where they can be packaged for display. These fragments are currently floating in the cytosol, but the display molecules are being built inside the endoplasmic reticulum, or ER. To bridge this gap, the cell uses a specialized transporter protein known as TAP. TAP acts as a dedicated pump, actively moving these short peptide fragments out of the cytosol and into the lumen of the endoplasmic reticulum. This step is a critical bottleneck in the pathway. If the peptides cannot get into the ER, they can never be displayed on the cell surface. Once the peptides successfully enter the endoplasmic reticulum, they are ready for the final stages of preparation. Inside the ER, these peptides are carefully loaded onto newly synthesized major histocompatibility complex class 1, or MHC1 molecules. You can think of the MHC1 molecule as the display tray from our factory analogy. It is specifically designed to hold these tiny peptide fragments securely. Once the peptide is firmly bound to this tray, the entire loaded MHC1 complex is packaged into a vesicle. This vesicle is then sent through the Golgi apparatus, which acts as a cellular post office, routing the complex toward its final destination at the cell surface. Following its journey through the Golgi apparatus, the loaded complex finally reaches the cell membrane. Here, the MHC1 molecule is securely anchored, proudly displaying the viral peptide on the outside of the cell. This is the glass window facing the street. Passing immune cells, specifically CD8-positive cytotoxic T cells, act as the police force. They constantly patrol the body and inspect these displayed peptides. If a T cell recognizes a normal, healthy cell protein, it moves on. But if it recognizes a viral fragment, it will immediately release toxins to destroy the infected cell, stopping the virus from replicating any further. Before we look at the mathematics of this process, let us appreciate why this pathway is so vital to human health. Because antibodies cannot enter cells, the immune system is completely blind to viruses hiding inside. If this pathway fails, the infected cell becomes invisible. For example, sneaky viruses like herpes simplex deliberately block the TAP transporter, allowing the virus to spread unchecked. Furthermore, modern medicine heavily relies on this exact mechanism. mRNA vaccines, like those used for COVID-19, work by instructing our cells to produce the viral spike protein internally. Our cells then use this very endogenous pathway to process and display the spike protein, successfully training our immune system. Now that we understand the biological steps, let us look at how we can quantify this process. The overall rate at which antigens are presented on the cell surface is governed by a specific formula. Rp represents this overall rate of antigen presentation, measured in complexes per minute. This rate is the product of 4 variables. First, Kc is the catalytic rate constant of the proteasome, telling us how fast the shredder works. Second, Cag is the intracellular concentration of the viral antigen, meaning how much raw viral protein is present. Third, E-Tap is the transport efficiency of the TAP protein, a fraction between 0 and 1. Finally, AMHC represents the binding affinity of the peptide to the MHC1 molecule. If the viral concentration increases, the presentation rate goes up proportionally. Conversely, if a virus mutates to lower the binding affinity, the overall presentation rate drops. Let us apply this formula to a concrete example. Suppose a liver cell is infected with hepatitis B. We are given that the intracellular concentration of the viral antigen is 500 molecules per cubic micrometer. The proteasome cleavage rate is 0.2 per minute. The TAP transport efficiency is 0.8, and the peptide's affinity for MHC1 is 0.5. We need to calculate the rate of antigen presentation. We start by substituting these biological values into our rate equation. First, we multiply the cleavage rate of 0.2 by the antigen concentration of 500, which tells us that 100 peptides are generated per minute by the proteasome. We then multiply this by the TAP efficiency of 0.8 and the affinity of 0.5. The final calculation yields 40 complexes per minute. Physically, this means every minute, 40 new viral peptides are successfully displayed, providing a strong signal for T cells. Building on our previous calculation, let us see what happens when a virus fights back. Imagine a mutated strain of the virus produces a protein that specifically inhibits the TAP transporter, reducing its efficiency from 0.8 down to 0.1. All other variables, including the amount of viral protein and the shredding rate, remain exactly the same. We need to calculate the new rate of antigen presentation. We plug the new TAP efficiency of 0.1 into our equation. The proteasome is still successfully making 100 peptides per minute in the cytosol. However, because the TAP transporter is inhibited, we multiply by 0.1 and then by 0.5. The new presentation rate is calculated to be only 5 complexes per minute. This creates a massive biological bottleneck, almost no peptides are making it into the endoplasmic reticulum. Now that we have found the new presentation rate, let us determine exactly how much the viral evasion tactic damaged the cell's warning system. We will calculate the percentage drop in presentation compared to the original rate. We use the standard percentage change formula, which involves taking the difference between the old rate of 40 and the new rate of 5, dividing that by the old rate of 40 and multiplying by 100. Subtracting 5 from 40 gives us 35. Dividing 35 by 40 and multiplying by 100 yields an 87.5% drop in antigen presentation. Physically, this means the virus has successfully hidden itself from the immune system by shutting down the supply chain of peptides. The infected cell is now almost invisible to passing T cells, allowing the virus to survive and multiply. Let us bring all these concepts and calculations together by looking at this comprehensive diagram of the pathway. On the left side, in the cytosol, you can see the folded viral protein. Follow the arrows as it enters the cylindrical proteasome, where it is shredded into small peptide fragments. Next, look at the membrane of the endoplasmic reticulum. You will see the TAP transporter acting as a channel, actively pumping those tiny fragments inside. Once inside the ER, the fragments are loaded onto the newly synthesized MHC class I molecules. Finally, follow the vesicle as it carries this loaded complex up to the cell membrane at the top right. The complex fuses with the membrane, leaving the viral peptide prominently displayed on the outside of the cell, ready to be inspected by the immune system. This visual perfectly summarizes the biological supply chain we just calculated. We have reached the end of our lecture, so let us review the key takeaways. The endogenous antigen processing pathway is your immune system's critical internal surveillance mechanism. It relies on a precise sequence of events. The proteasome shreds dangerous intracellular proteins. The TAP transporter moves those fragments into the endoplasmic reticulum, and MHC1 molecules carry them to the cell surface for display. As we saw in our calculations, any disruption to this pathway, such as a virus blocking TAP, drastically reduces the presentation rate and hides the infection. This continuous sampling and display process ensures that any cell harboring a virus or turning cancerous is quickly identified and eliminated by cytotoxic T cells. You now have a solid understanding of how our bodies fight off internal threats every single day.